Hello, we're working through the last unit here. So we have a lot of overlap now with DNA structure and RNA stuff like that. So that's kind of how the year goes. After basic stuff happens, all the rest is application. But here we're gonna just hit transcription and a little bit of RNA editing, and I'm gonna chop this up in different ways. This last uh, DNA gene regulation unit. So here we go. So learning objective. Describe the mechanisms by which genetic information flows from DNA to RNA to protein. That's the central dogma, right? But you got to add trait here. That's a key thing. See what it says here, right? This isn't in the blue box. These are one of these suggestions, kind of hints like the misconception sheets that you need to read, right? Students failed to provide reasoning connecting a change on the molecular level, change in DNA is what they're talking about, to a change in phenotype. That's what trait is. They're saying that you're not doing a great job making this connection, which is pretty easy. So do a great job, right? This is all about the central dogma, right? So anyway, talk about, genes causing traits, and you skip the middle part, and that's what they're trying to tell you here. So anyway, genetic information flows from a sequence. Information is all about sequences, just like it is in this blue box. It's sequence of alphabet letters, right? Information flows from a sequence of nucleotides, more specifically their nitrogenous bases, A, T, C, and G, in DNA to a sequence of bases in RNA to a sequence of amino acids in protein, and it's proteins by and large that cause traits. All of these are talking about the central dogma. Okay. So now they got some stuff here that I'm going to have to call them up and change, because I guarantee you they're going to change a couple things in here. So let's take a look, right? This is transcription. This is the step in the central dogma, right, where DNA is used to make RNA, right? This is not a chemical symbol arrow that means DNA is changed into RNA, so you don't want to say that. DNA is used as a template, a pattern to make RNA. A pattern of what? Nitrogenous bases attached to part of a nucleotide, right? So here we have the enzyme involved, only one this time, unlike 5 different ones in replication, just RNA polymerase, right? So uses just one of the DNA strands. They put that word single in there on purpose, right? To direct the inclusion, I wouldn't use that word, right? To direct the addition of nucleotides that makes an RNA molecule with a specific base sequence that is complementary to that one in DNA. Okay, so here we have the RNA polymerase, right? And here we have the idea that only one strand is getting used. This, I don't know why they put in this. I've never liked this. I understand it kind of why they chose these words, but I think it's like backwards to me. So, but they're in the blue box. They're not going to ask you what it means, but they might use these terms. So here we go. The DNA strand acting as the template. That would be this one down here, right? It's the one that's being used to put together this RNA. Is referred to as the non-coding strand. It almost like makes opposite sense. I would call it, this is the one that we're using the code of this one to make this one, right? And I do understand why they called it this, but just so you know and not be confused, the minus strand, I have hardly ever seen that. I don't know why they chose to put this in here. The antisense, that almost doesn't make sense, pun intended, right? Because this is the strand that's actually being used. It actually kind of like makes sense. But selection of which of the 2 DNA strands serves as the template, that has to do with a lot of factors called transcription factors and what that we'll get to in a later video. So here we have, just wrapping it up, because this is really short. This is the simplest, kind of thing. You have this DNA. You have this part of a much longer chromosome, much longer, literally 1,000 more, way more than 1,000 times longer, if this is one of your chromosomes. And this one section is the only section we're going to unzip and use. And then that, as a matter of fact, we're only going to use one side of that section. but it's this section that we most commonly refer to as a gene, right? It starts with an area that says start here. Promote means start. That's why that's called a promoter. That did not make it into this part of the syllabus. I think it will later in the regulation part, right? And it's got a part that says, okay, you can stop here, right? So this is like, to make our analogy, a chapter, a recipe, so to speak, not a chapter, but a recipe in a recipe book to make one thing. What one thing? This completed RNA, which because it's transcription that's doing this, we call it a transcript, right? So note here the differences with replication compared to this because there's a lot of similarities. It's in the nucleus, right? Unless you're a prokaryotic cell, then it's just happening in your little body, right? It's in the nucleus. DNA unzips, but here DNA zips back up and we're done, and it's no different than it was when we started. It starts at a place called the promoter. This unit we call the gene, and the product here is that RNA that we can also refer to with the word transcript. Excuse me. So here's a little editing stuff that I added in this part, right? And they pretty much spell it out for you right here. We've gone over all this stuff. In eukaryotic cells, so this is not a prokaryotic thing, the initial messenger RNA transcript, we would call that this, the initial transcript, the pre-messenger RNA, they call it here. You get the idea, right? It's going to have some parts cut out. What are the parts that get cut out? The parts that start with the word in, which I always thought was confusing, but what it stands for is, again, intravening, in between, intravening, the parts that aren't going to get cut out. And those are the parts that are going to get used, even though the prefix X kind of means out of, since these are the parts that are going to get expressed, that's where the X comes from. Intravening is where the N comes from. And this is all also about gene expression, which is also about the central dogma. If this gene is made of the chemical DNA, and the expression of it finally is seen as a trait, right? Then the first step in that gene expression, in fact, the only step that the gene is actually involved in, is this step of transcription that we're talking about, right? Then in translation, we're going to make that protein that causes a trait. Okay, so they talk about all this, and they say also that a bunch of adenine nucleotides is added to one end, called simply a poly A tail. A modified guanine triphosphate is added to the other end, the 5N specifically, so that's called a 5' cap. Notice those terms, that term 5' cap is not in here, but you might bump into it. So this is the idea, and then this last thing, excuse me. If you took this primary transcript and you cut out different parts of it and put together different parts, that can generate different versions of the resulting mRNA molecule known as alternative splicing. This is also part of gene regulation. At one point in time, a cell chops it up and puts it together to be like this to make a particular protein. And at a later point in time, it chops it up differently, takes out different introns, puts together different exons to make a different protein, which causes a different trait. So we'll get back to that again later. This I threw in here because I could put it in a couple places, but since this is about transcription, and this thing is the opposite, reverse transcription, catalyzed by an enzyme aptly named reverse transcriptase, this is a kind of molecule that we have borrowed from bacteria cells in genetic engineering, so we'll come back to it. But this is the kind of thing that these creatures called retroviruses have evolved to be able to have and do, the ability to take their RNA and do the reverse of that normal dogma thing and use it as a template to make DNA. So that's where I stuck that in, and we'll come back to it, I think, in genetic engineering stuff. So see, this DNA is trying to get into the nightclub. You get the idea. So I've been to this nightclub before, and I was sitting at the bar, and Descartes walked in, and he had a beer. You know Descartes, you know that philosopher guy, and he had a beer, and the bartender says, "You want another?" He said, "I think not," and then he disappeared, never to be seen again. You might have to think about that one.